Assalamualaikum. Let's talk about protein synthesis. As you already know that the protein synthesis begins with the formation of peptide bonds. So how peptide bonds are formed? Let's look in an animated view. In this activity, we will examine biochemistry behind the formation of peptide bonds. Inside the cells, peptide bonds are formed within ribosomes during a process called translation. So, translation is basically a process by which the peptide bonds are formed within an organelle called ribosomes. So, proteins are polypeptides, we all know, that are folded into specific three-dimensional structures. So, proteins are three-dimensional structures formed by the polypeptide chains. Polypeptides are composed of amino acids covalently bounded to each other like beads on a string. So, polypeptides are formed by the amino acids which are like beads on a string. The covalent bond that holds amino acids together is called a peptide bond. The white line shows the peptide bond between different colored amino acids that are forming a polypeptide chain. And when this chain is folded into a three-dimensional structure, it forms a structure called protein. So, all 20 amino acids share a common structure that includes a center alpha carbon that is shown with the red color covalently bounded to a carboxyl group yellow color that is COOH, an amine group NH2, a hydrogen molecule H with a green color, and a variable R group. So the basic structure of all amino acids, 20 amino acids, is same except the R group that changes and makes the amino acids differ from each other. So there is a central alpha carbon that is red in color, the carboxyl group COOH, and the amine group that is in blue color NH2, and the green color hydrogen molecule, and the R color variable R group. So, study the three amino acids below. Can you identify the central alpha carbon? Yes, this is the central alpha carbon. This is the carboxyl group. Can you identify the amine group? That is the blue color amine group. Can you identify the R group? That is different in all three amino acids that make them different. Alanine has CH3, while cysteine has CH2 and SH, while serine has CH2 and OH. So all other the basic structure is same except the R group that varies and make each amino acid different. So during a protein synthesis or translation, amino acids are covalently bounded to each other through a peptide bond. This section, which takes place within the ribosome, is known as dehydration synthesis reaction. Dehydration because the loss of water molecule, and synthesis because it's built. Dehydration synthesis reactions are commonly used in cells for anabolic or synthetic reactions. Watch this formation of a peptide bond between two amino acids carefully. The OH is removed while H is removed from amine group of the amino acid on the right. The OH and H form water. This is dehydration process. A covalent bond can now form between the amino acid. This is the peptide bond. So let us look at again this section. Look it carefully. The hydroxyl group is removed from carboxyl group of one amino acid that is left, and from the right amine group H is removed, that is water molecule is removed, that is dehydration process. Now a peptide bond will form between the carboxyl group and the amine group of the second amino acid. So this is the peptide bond formation. I hope you had understood it. Now go further. You have completed this activity of peptide bond formation, which leads to the protein synthesis. So, for more videos related to this topic and for chemistry topics, do subscribe to Subject Expert. Thank you.